Hi everyone, my name is Megan Beaudry and I'm a third-year PhD candidate working under the advisement of Dr. Travis Glenn at the University of Georgia. Today I'll be presenting on some of my master's thesis work, which I did at the University of Alberta. The presentation I'll be presenting today is called From Nuisance to Resource, Understanding Bacterial and Microbial Sources of Contamination in Urban Stormwater-Impacted Bodies of Water. So first, I'll go over why we use stormwater. Then we'll look at how we sampled and analyzed the water. And finally, we'll look at some of the results on water quality, microbial source tracking, and enteric bacterial pathogens. Then we'll conclude with what all of this means. So many of you may be wondering where in the world Alberta is. Alberta is a province on the west coast of Canada. In this province, I work here represented by this nice Oilers logo. And this is where the University of Alberta is in the city of Edmonton. Down to the south of us, approximately 4 hours away by car, is the city of Calgary, represented by the Flames logo. So currently, cities in Alberta, such as Calgary, are looking for ways to address their growing demands on water resources. Now, the demand on water is increasing for two main reasons. First, climate change, and second, because the population of Alberta is growing, because everyone wants to live someplace where 10 months of the year is winter. And so stormwater is precipitation that is collected after reaching the ground, which is a key difference between stormwater and rainwater. Our study focused on urban stormwater, which is runoff that is created by the increase in impervious surfaces that occur in urban environments. The purpose of stormwater ponds is to collect excess runoff from storms in cities in order to control flooding and manage urban pollution. Therefore, stormwater, which was originally seen as a nuisance, is now seen as a potential resource. And so we can reuse stormwater in a variety of ways, such as watering community gardens or athletic fields, toilet flushing, clothes washing, firefighting, and water features. Stormwater reuse poses a variety of challenges due to the potential of this source to be contaminated with human and animal feces, and thus microbial pathogens, such as Campylobacter, Salmonella, and pathogenic E. coli. The contamination of water with human or animal feces poses a significant risk to human health, albeit the risks associated with pathogens found in human waste are far greater than the risks associated with animal waste. Several recent studies have demonstrated that human feces are commonly found in urban stormwater systems, and therefore urban stormwater risks associated with its use must be better understood in terms of contamination sources. And so I wanted to identify sources of bacterial contamination and bacterial pathogens in stormwater ponds located in Calgary, Canada. All of our samples were taken through the GRAB sampling method. 3 stormwater ponds were sampled in the city of Calgary, with the number of inlets and outlets ranging from 4 to 5. Each pond was sampled twice a week over 21 weeks, resulting in 533 samples. All of these 3 ponds were investigated. However, due to time constraints, we're going to focus on one today, which is called McCall Lake. In order to determine the sources of contamination in the stormwater pond, both molecular and culture-based methods were used. Immediately following water sampling, the samples were sent to the closest provincial laboratory to be filtered. 20 mils of the sample was filtered through a 0.4 micron filter, in which DNA was extracted using EPA method 1611. This is a bead beating method to DNA extraction. An additional 100 mils of the water was used in quantitrays to detect E. coli. DNA was then extracted from E. coli positive wells. Following DNA extraction, qPCR was performed. And so in essence, this project has 2 stories. The first story is the story of the contributors to the pond. So what animals are in our urban environment? Well, several qPCR targets were run, which are Bacteriodes-specific microbial source tracking markers. We have 2 markers for humans, 1 for ruminants, 1 for dogs, 1 for Canadian geese, 1 for muskrat, and 1 for seagulls. All of these, except for HUMM2, are based on the 16S ribosomal RNA targets. Furthermore, they're all targeting bacterial species that are associated with that particular host. The sensitivity is as low as 10 copies or less per qPCR reaction. So the second story is what is in this pond that could be a risk to human health. And this is where we use targets for pathogens and indicator organisms. The only indicator is Entero-1, which detects Enterococcus. We use the NVA target for Salmonella and the Van Dyke targets for Campylobacter jejuni, coli, and Laurii. We use Shiga-toxin 1 and 2 for each of the Shiga toxins in E. coli, and heat shock protein 60, which targets Arcobacter Beaudry, which is a putative pathogen. For culture methods, we performed 2 separate assays. We performed 1 assay for culturing for Campylobacter and 1 assay for culturing for Arcobacter. These are most probable number qPCR assays. Now, there are 2 main purposes for the use of these assays. First, I'm culturing the bacteria to tell if they're culturable or alive. This allows us to quantify the number of bacteria. This quantification will aid in the determination of risk. Secondly, the purpose of culturing is to build a library of isolates that will be tested for the potential of virulence genes using different primer sets. Now again, the method that was used was a most probable number qPCR assay. For this assay, water samples are centrifuged within 24 hours of sampling. These samples are then aliquoted into 96 well plates and allowed 48 hours for growth at 32 degrees for Arcobacter and 42 degrees Celsius for Campylobacter. Following growth, the bacteria are enumerated by qPCR. So here's an example of a stormwater pond. This is McCall Lake. 4 sites were sampled within McCall Lake. We have 2 outfalls, which is where water flows into the pond. So we have outfall ML1 and outfall ML2. Outfall ML2 is above ground, and you can see here that outfall ML1 is partially submerged. We also have 2 inlets. Inlets were defined as where water leaves the pond. And so these 2 tables here compare the makeup of our 2 outfalls. As represented here, the catchment area for ML1 is approximately 1,500 hectares, comprising residential, major infrastructure and transportation, industrial, parks and institutions, commercial, and future urban development land uses, as noted in our table over here. So this is a very mixed area, and so the sources of contamination would probably also be mixed, with things from roads, neighborhoods, parks, which could mean dogs, will also be mixed. So, however, ML2 tells a slightly different story, as this is mostly comprised of industrial area. And so this graph represents the occurrence of Campylobacter, Salmonella, Arcobacter Beaudry, and Shiga toxin producing E. coli in stormwater over 553 samples tested by molecular methods. As represented here, Arcobacter was much more prominent than all our other pathogens in almost 25% of samples. So we did a comparison between our culture and molecular methods to allow us to see how accurate our molecular methods really are. And we are confident in our molecular methods for Campylobacter due to the fact that when we culture for Campylobacter, we also cannot find it. However, for Arcobacter, culture and molecular were both positive 6 times out of 32. However, there are 18 instances where we are able to detect Arcobacter through culture, but not through molecular methods. This leads us to believe that the previous slide may be even an underestimation or a conservative estimation, since 25% of samples contained A. Beaudry. However, this may be as high as 45% based on the results of our culture. This is so we can see the dominant source of pollution in the system based on the frequency of detection of any of the markers above the threshold. All targets have been validated to have similar overlapping limits of detection. Human markers are more frequently detected than other markers in the system, with it being the dominant source of pollution being HF183 in approximately 27% of samples, and HUMN2 in 10% of samples. The only other dominant source of pollution is coming from birds such as seagull at 9%. Third, we noticed that the other source tracking markers, so Canada goose, dog, muskrat, and ruminant, were detected in less than 2% of samples. We also noticed that the frequency of human fecal contamination varies between the individual ponds sampled, with 39% of samples in McCall Lake detecting human fecal contamination. So we then ask ourselves the question, is there one site within that pond that drives up this frequency of human fecal contamination? And here we can note that ML2 is frequently contaminated with human fecal contamination, with 93% detecting human fecal contamination at that sampling site, ML2. This claim is further substantiated by the fact that HumM2 is also more frequently detected at sampling site ML2 in 59% of samples. And so here our purple line represents the number of copies of HF183, which is again a Bacteriodes specific marker for human contamination. Over the summer, HF183 was consistently seen, however, at varying levels. There's a gross trend regarding E. coli levels, which are represented by the orange line, and HF183. As represented here, in many cases, as HF183 spikes, so does E. coli. And when E. coli increases, HF183 also increases. To further back the evidence of human contamination and the results that are not due to cross-reaction are the results of HumM2. HumM2 is another Bacteriodes-specific marker for human fecal contamination. It's not as sensitive as HF183. However, when HF183 reaches levels above log 3, HumM2 is also found. Again, there is a potential for human sewage to be associated with our E. coli numbers at this outfall. So this graph overlays the levels of Arcobacter Bootsleri with two markers for human fecal contamination. Temporal variability was observed in these samples for Arcobacter throughout the stormwater season. We also note several times where the levels of HF183 and Arcobacter increased together. And so we think there might be a relationship between human fecal contamination and Arcobacter Bootsleri. However, there are several times where Arco is detected in the absence of human contamination, leading us to believe that another source of Arcobacter Bootsleri may be present. So now we're going to briefly discuss another outfall in McCall Lake, ML1, which has a much lower frequency of human fecal contamination. As a reminder, ML1 was the outfall that was made up of mixed areas, so industrial, residential, transportation, and parks and rec. Here our blue line is LeeSg, which is a qPCR specific marker for seagull, which was our second most dominant marker behind ones for human fecal contamination. LeeSg was seen sporadically throughout the season, as would be expected, and at times with Arcobacter. So even in the absence of human heavy markers, we also saw a spike in LeeSg associated with Arcobacter. So Arcobacter Bootsleri is highly dominant in wastewater, and our lab has actually done some research on this area in which a manuscript is being prepared. Our own data support this, that we may find levels up to 10 to the 6 per 100 mil in raw sewage. However, we do not believe that this is the whole story regarding Arcobacter. We detect Arcobacter in the absence of human markers, as I showed in the previous slide. We can even occasionally see it with birds. Further, Arcobacter is unique for its ability to survive and replicate in the environment. Therefore, we suspect that there are multiple influences, such as wastewater, birds, and endogenous growth. So as a reminder, Arcobacter Bootsleri is a relatively new genus that was derived off of Campylobacter 20 years ago. It is a putative pathogen. It is a relatively new genus that is known to be more hardy than its weaker sister Campylobacter. Due to the unknown nature of this organism, we sought to isolate it to determine its virulence potential. Homologs of virulence genes in Campylobacter were chosen to test against isolates from McCall Lake culture samples. So of these 85 isolates, 73 were determined to be unique by qPCR, which distinguishes them based on unique banding patterns and peaks in an electropherogram. qPCR is a method of bacterial fingerprinting, which identifies an imperfect palindrome. It is a similar concept to rapid PCR. However, qPCR was found to be more reproducible and complex fingerprints than rapid PCR. And so we tested 9 different virulence genes listed here. I screened for the presence or absence of these genes in the A. Beaudry isolates by looking for a banding pattern on a gel after running endpoint PCR in 73 unique isolates. And our results show that we have a potentially virulent pathogen. There were 5 virulence genes that were present in more than 90% of samples, and 1 present in all of them. To try to further determine if virulence was associated with a source, I overlaid the presence of virulence genes with microbial source tracking results. However, no consistent relationship between fecal source and virulence marker was found. And so what are our findings? Well, human sewage appears to be a dominant source of fecal pollution in urban stormwater systems. Arcobacter Beaudry was the most prevalent bacterial enteric pathogen found in stormwater in approximately 25% of samples. Arcobacter Beaudry sources likely include humans, animals, and growth in the environment. And so what does this mean? Well, at this stage in the project, the water is not considered a pristine resource. Research is still continuing by examining chemicals, helminths, and potential viruses that could be present in the stormwater ponds. Further work includes characterizing and addressing infrastructure challenges associated with human fecal pollution, such as the one we see at sampling site ML2 located in McCall Lake. We also want to assess the occurrence of other pathogens in the stormwater. I'd like to thank the Provincial Laboratory for Public Health for their assistance in this project, in addition to all my mentors and coworkers at the University of Alberta School of Public Health. I would also like to recognize our funding agency, Alberta Innovates, for their funding for this project. Thank you.